Hi, my name is jbharj, and I did my presentation on antibiotics in food animals. First of all, antibiotics are drugs that are used to fight bacterial infections. They're also known as antimicrobials and were first discovered in 1927 by Alexander Fleming. Penicillin was the first particular antibiotic that he discovered. Their widespread use started in the 1940s and dramatically reduced illness and death from infectious disease. The discovery of antibiotics is known as one of the major milestones in public health history. What is antibiotic resistance? Antibiotic resistance is the ability of bacteria or other microbes to resist the effects of an antibiotic. Listed among the CDC's top concerns, antibiotic resistance has been called one of the world's most pressing public health problems. At this point, almost every type of bacteria has become stronger and less responsive to antibiotic treatment due to the widespread use of antibiotics today in our society, not only for medication purposes to fight infectious diseases and bacterial infections, but also because a lot of antibiotics are used in foods to help animals fight bacterial infections and diseases, and then we eat those animals, and therefore that resistance is then transferred to us. The increasing spread of resistance is an outcome of natural selection and can be viewed as an expected occurrence of Darwin's principle of survival of the fittest. In any large population of bacteria, a few cells will be present which possess traits that allow them to survive in the presence of a noxious substance, i.e., an antibiotic. These cells lacking those cells lacking this beneficial trait will be eliminated, leaving only the resistant bacterial behind. Basically saying that the ones that have the most beneficial traits will survive and the ones that don't have the beneficial traits will die. And that, once again, is the same thing that Darwin said about animals. And that's how he said that evolution occurs. So basically, that's how we become resistant to bacteria, because the bacteria that is resistant survives and the bacteria that's not resistant dies. And then once again, that the one that is resistant to the antibiotics keeps multiplying, and they become increasingly more prevalent in our bodies and in society in general. Long-term antimicrobial use will dramatically change bacterial populations, with more resistant organisms increasing in proportion. This can result in a situation where the next time an antimicrobial is needed, it may not be effective to treat what was once an easily treatable infection. Mechanisms of resistance. Bacteria can acquire resistance through mutation. Antibiotics affect bacteria by binding to and inhibiting specific enzymes and facilitate DNA replication, which ultimately destroys the bacteria. However, when a genetic mutation occurs in which the area in the area of coding, gene coding, it keeps bacteria from binding efficiently, which allows DNA to continue to replicate. Another way that resistance occurs is through destruction or inactivation. Here, antibiotics are either degraded or modified by enzymes in the bacterium, rendering them ineffective before they reach their target site and damage the bacterial cell. So, therefore, the bacteria themselves have an enzyme that destroys the antibiotic before it can even take effect. Efflux pump is a channel that actively exports antimicrobial and other compounds out of the cell in order to prevent an intracellular accumulation necessary to exert their lethal activity. Basically, this is saying that before the antibiotic can accumulate within the cell and destroy it, there's a pump that just pumps the antibiotic right out of the cell, and therefore it never gets a chance to actually do what it's supposed to do. Genetic transfer is another way bacteria can acquire resistance. Conjugation is one means of genetic transfer. It's mediated by a circular DNA called a plasmid. The plasmid actually has the gene which is resistant, which makes the bacteria resistant. to the antibiotic. This plasmid then replicates within the cell, within one particular bacteria, and then when another bacteria is really close to the bacteria that has the plasmid with the resistant gene, they form a bridge-like structure that allows the copied plasmid in the first bacteria to be duplicated and transferred to the other bacteria. Therefore, the resistant gene is then transferred from the first bacteria to the second bacteria. Transformation is another form of genetic transfer. When cells die and break apart, the DNA can be released into the surrounding environment. Other bacteria in close proximity can then take up this DNA and incorporate into their own DNA. This can lead to resistance because if one of the genes in the bacteria that died was resistant to a specific antibiotic, the new cell that took up that DNA can now also acquire that resistance. Transduction is the third way that genetic transfer can occur. When a virus infects a bacteria, it takes over the bacteria's genetic processes to reproduce. During this process, the bacterial DNA is incorporated into the new virus's DNA. When the virus breaks out of the host bacteria and goes on to infect other bacteria, it can then transfer that gene that was copied into its DNA from the first bacteria into the new bacteria that it infects. Antibiotic use in food. The two main ways that antibiotics are used in food are therapeutically and subtherapeutically. They're used in feed to improve gut health and limit subclinical diseases, which translates to more rapid and efficient growth. So basically, farmers use antibiotics in order to not only treat animals that are sick, but also to keep animals from getting sick so that they grow better and grow faster. Therapeutically, antibiotics are used in high doses for short periods of time. These antibiotics are used to treat specific diseases or infections. So if an animal is sick, you will use a therapeutic antibiotic to treat it. Subtherapeutically, antibiotics are used in low doses for a longer period of time. These are used to prevent infections and improve growth rates. So subtherapeutically, antibiotics are used to prevent diseases and to get animals to grow faster and more efficiently. Antibiotics use in food is also estimates of antibiotics use. The Animal Health Institute estimates that 18 million pounds of antibiotics are used in beef, dairy, swine, and poultry. 83% are used for the treatment of specific diseases. The Union of Concerned Scientists states that 25 million pounds of antibiotics are used for subtherapeutic uses alone. So our farming industry, our agriculture industry is using a lot of antibiotics to treat animals. It's not just every once in a while. So that's why it's such a main concern. So what's the big deal? Well, antibiotics are bad for humans because they, once again, cause antibiotic resistance. And this can cause significant danger and suffering for children and adults who have common infections that were once easily treatable with antibiotics. This can lead to increased and prolonged hospital stays and medical expenses, as well as death if infections are not caught and treated in time. Antibiotic resistance is also bad, or excessive antibiotic use, rather, is also bad for the environment because nearly 2 trillion tons of animal waste is produced each year in America. This waste contains significant amounts of undigested antibiotics and antibiotic-resistant bacteria, which can contaminate surface and groundwater, harm natural ecosystems, and eventually make their way back to humans. Antibiotic use is also bad for livestock. Animals on factory farms are given antibiotics to compensate for their overcrowded and unsanitary living conditions and to help them gain weight. Livestock allowed to lead natural, healthy lives are healthier and need fewer antibiotics. So farmers basically compensate for keeping animals confined in small, dirty, unsanitary, crowded areas where they catch diseases from one another by using lots and lots of antibiotics. But once again, that's just a vicious cycle. So we prefer that animals lead more natural lives in which they don't get diseases in the first place so that they don't need to be treated with antibiotics. So, what is being done? The U.S. Government Accountability Office recommends, in light of the emergence of the antibiotic resistance in human, questions about the extent that the agriculture use of antibiotic contributes to human health burden, and the debate over whether further regulations or restriction of use in agriculture is needed, we recommend that the Secretaries of Agriculture and the Health and Human Services develop and implement a plan that contains specific goals, timeframes, resources needed to evaluate the risks and benefits of the existing and future uses of antibiotics in agriculture, including identifying and filling critical data gaps in the research. So, this is basically telling us that um, the U.S. government's accountability office is saying that we need to figure out a way that antibiotics are used and how they can be helpful to us, and do the research and establish timeframes and methods and study what's going on in order for us to make good judgments. The CDC's Get Smart program is a program getting the general public to be educated on how antibiotics are used and how they work. So it's an education initiative aimed for people to become well informed about antibiotics so that they can make good decisions in their everyday life. And finally, the World Health Organization is strongly urging all countries to take measures to monitor and control the use of antimicrobials by focusing control and prevention efforts in four main areas: surveillance for antimicrobial resistance, rational antibiotic use, therefore, educating healthcare workers and the public on the appropriate uses of antibiotics, and introducing or enforcing legislation related to stopping and selling of antibiotics without prescription, strict adherence to infection prevention and control measures, including the use of hand washing measures, particularly in healthcare facilities. So, we want to prevent people from overusing or abusing antibiotics so that way, when we do really need them, our bodies will be able to take full advantage of them. And the last slide is my references, and that's it. Thank you.